today we are going to read the next topic from the cell, the basic unit of life, that is about the locomotory organ. Now, the locomotory organ in case of the prokaryotic organism and eukaryotic organism, it is being found that the two common type of locomotory organs which are found, that is, one is your cilia and another one is flagella. The basic structure, ultrastructure of cilia and flagella is the same. In case of the prokaryotic flagella, it is made up of the protein which is called flagellin, whereas if you talk about the eukaryotic flagella is made up of tubulin. Now we are here going to read about the ultrastructure of cilia or flagella, because both are same, as I told you, in terms of the eukaryotic cell. Now the flagella is a locomotory organ which can be of two types. It has been noticed, one is whiplash type and one is your tinsel type. The tinsel type of flagella is having some hairy outgrowths on it, whereas the whiplash type is the sheath of the flagella is smooth. And if that is the origin or the basal body of the flagella is noticed, it is somewhat similar to that of the centriole. We are going to study about centriole after this flagella only, but today we are going to concentrate at first in the flagella or the cilia. So as I told you that since it's a eukaryotic cell, flagella, so it is made up of tubulin protein. Every flagella, if we see the ultrastructure of the flagella, then it is surrounded by a common sheath which is similar to like that of the cell membrane. Okay, and inside it is the axoneme which is comprising of your total of 11 microtubules. Out of this 11 microtubules, 2 are the central microtubules which are again surrounded by a central sheath, and there are total of 9 peripheral tubules which are arranged. Now this type of arrangement is known as your 9+2 arrangement. Okay, now the peripheral tubules which has been noticed. Now this peripheral tubules in turn is made up of 2 subunits. Okay, the central protein which is present surrounded by the central sheath is made up of a protein which is nothing but a ATP synthase. Okay, it is called ATP synthase enzyme. It helps in the hydrolysis of ATP, and that energy is actually utilized for the contraction of the peripheral 9 proteins, and as a result of it, it is a united effort to be seen in the form of movement of the organism. That is, the flagella's movement is uniform because of the contraction of the 9 peripheral proteins assisted by the ATP synthase which is present in the central that is your 2 subunits. And these peripheral proteins, the 9 peripheral proteins, are in the form of doublet. That is, we have indicated them as your A and B, and A is made up of again subunits, 13 subunits, and B is made up of your 10 subunits. And A is producing 2 arms. Okay, and they are producing 2 arms. One of the arm is adjusted or is attached to that of the B, and that is how it is a united structure. And from A again a spoke is arising with a globular head, and there is a transition junction which is connected to that of the central sheath of the 2 subunits which is in the center. So this is what is known as your 9+2 arrangement of the ultrastructure of flagella or cilia, which can be seen under the electron microscope. So this structure is very important from examination point of view. You have to practice it with the proper labeling in the exams if it comes, so that you can draw it as well as label it perfectly. After the ultrastructure of flagella and cilia, now we are going to study about another organelle, very important organelle in the animal cell, that is called centrosome. Now a centrosome is made up of 2 centrioles. Now the centrioles is perpendicular to each other, and they occupy a space which is not bounded by any kind of membrane. No unit membrane is present around the centrosome, and these that is centriole helps in the formation of the spindle fiber during the time of cell division. Now the centrosome centrioles which are perpendicular to each other, they are known as your diplosome, because they are 2 in number, so they are known as your diplosome, and they occupy a certain space near that of the nucleus, and that is known as the centrosphere. The area is known as centrosphere. So centrosphere together with the diplosome is known as your centrosome. Okay, now the centriole in turn, if you see it closely, this is the that is ultrastructure, or the structure in the electron microscope. How does the centriole looks like? Now the centriole has the 9 peripheral that is your microtubules. Okay, so 9 that is peripheral microtubules, and they, unlike that which we have seen in case of flagella having the 2 central tubules, they do not have a central tubules, they have a central hub, and to this central hub, from each of this microtubule, 9 spokes, which is again made up of protein, comes out and it joins with that of the central hub. So this type of arrangement is known as your 9+0 arrangement, which we have seen in case of your flagella. It was 9+2 arrangement. In the center there were 2 microtubules, but here there is no microtubule in the center. There is only a central hub and the peripheral that is microtubules. Now this microtubules, if we consider this to be a unit, these are the units. So microtubules in turn is made up of 3 subtubules. Now this subtubules in turn is made up of the protein, which is known as your tubulin. Okay, so the protein is known as tubulin, and the 3 subunits, as we have seen in case of your flagella, it had 2 subunits, and we had named it as A subunit and B, and the 2 adjacent that is microtubules were connected with the help of some linker protein. That is, A was connected with that of the B. Here the A is connected with that of the C. So the linker is there, but here the linker is between A and C, unlike A and B, which we have seen in case of your flagella. So this is known as your connector, and this is also proteinaceous in nature, and A is connected to that of the C. So this is the arrangement of that of your that is your centrioles in the cross section, and this model is known as your cartwheel model. Okay, that is the bullock cart's wheel, so it somewhat looks like that, so that is why the name has been given as your cartwheel model. Okay, now the purpose of centriole, I have told you, that is it takes part during the time of cell division in the splitting up of the that is your sister chromatids to the 2 daughter cells. Also, I have told you when I was teaching you about the ultrastructure of flagella or cilia, that the basal body of the cilia or the flagella is made up of centriole-like structure. So that means if we see the basal body of the flagella or the cilia, there also we will find this type of structure. But when we come towards the other end, that is the upper end, then the arrangement there is 9+2 arrangement. So there were total of 11 microtubules, and here there is only 9 microtubules, and in the center there is no microtubule. Okay, and these that is the subunits in turn is made up of some protofilaments, and as we have come to know that in A's that is subunits there were 13 filaments, and in B there was 10. So similarly here also A has 13 subfilaments, and the B and C is having 10. Okay, so this is all about your cartwheel model of centriole and the function of centriole. The 2 main function: it forms the basal body of the flagella or the cilia, and also it helps in the formation of spindle fiber so that the sister chromatids can be pulled apart during the time of cell division. So these were the 2 things which we have discussed today. I hope you have understood it well, and also the prior classes in which we have discussed cell in general, the difference between plant cell and animal cells, cell wall, cell membrane, and the various the 2 organelles which separately I have dealt with, that was your lysosome and mitochondria. See those videos again if you have any confusion, and then this chapter is totally complete, and we are now ready to go for the next chapter. Okay, so that's all for today's class.